Hello, you guys. In this video, we are going to wrap up the Unit 1.3 notes by looking at the last topic, Topic 3, which is actually the very last topic of all of Unit 1. So we finally made it. In these notes, we're looking at enzymes. And in Topic 3, we're going to end all of this by looking at how enzymes are being regulated inside of living things. And so it's really important now to take a moment and talk about and look at different ways that enzymes are being controlled inside of cells. So what's not happening right now in your bodies, you guys, and in living things, right now what's not happening is you don't have all these enzymes that you're making inside of all your different cells. And right now, all those enzymes are on and working and catalyzing all these different reactions at every moment of every day, at any specific time. That would be chaos, okay? And in living things, we're actually, and in cells even, it's very, very controlled and organized what's going on. And at any time or situation, there's certain enzymes that are being used, there's certain enzymes that are being used a lot, there's certain enzymes that are not being used, and that changes and fluctuates day to day and moment by moment, and depending on what's going on inside those cells and inside the environment of those organisms. And so this is all called regulation. Regulation is referring to how living things are actually controlling all of the stuff that's taking place in a way that's very orderly and not chaotic. Because the last thing we want is for all your enzymes in your body right now to be catalyzing all the chemical reactions they're able to catalyze. That would be a disaster. You would be dead. Right now, there's certain enzymes being used in certain cells, and there's certain enzymes not being used in those cells, and that's going to change moment by moment. And so let's look at how that takes place. So the first thing I want to talk about is something called allosteric regulation. And so basically, fundamentally, the way that these enzymes are being controlled is by utilizing the fact that these enzymes, which are proteins, they have two different conformations. They have an active conformation, and then they have a conformation that's inactive. Conformation is referring to the shape. So enzymes, they have a certain shape that they take on when they're active, and they're using their active site that's binding to the substrate and catalyzing chemical reaction. But then those same enzymes can change shape in a way where the active site is no longer able to bind to the substrate, and then it's not catalyzing those reactions. So think about the enzymes as having the ability to turn on or off depending on if they're in their active conformation or their inactive conformation. And so there's this thing called allosteric regulation, which is when you have a molecule binding to a protein, and when it binds to that protein, it changes the shape of that protein at a different location that it wasn't binding to. And so basically you have this protein, this blob, and then something binds to it, and then it causes this area over here of the protein to change shape because something binded to this other area of the protein. And so that's what's happening in allosteric regulation. Now, and specifically to enzymes, let's just make this easy. For enzymes, what we're going to look at in a moment when we're talking about allosteric regulation is we're talking about how there's going to be these molecules, we call them regulatory molecules, that are going to bind to enzymes at a location that's not the active site. So they're going to bind to the enzyme somewhere that's not the active site. But by binding to that location, they're going to change the shape of the active site. That's what allosteric regulation looks like when we're looking at enzymes. Now, this can be inhibitory or stimulatory, meaning that this depending on the molecule, this could either activate or stimulate the enzyme and start making it go into its active conformation and catalyzing reactions, or this could be inhibitory and could cause the enzyme to take on its inactive conformation and then slow down its ability to catalyze reactions. So let's start with activation first. So allosteric activation, this is a situation where there's going to be a molecule binding to an enzyme, and it's binding to the enzyme at a location that's not the active site. So it's binding in this picture here, this green triangle, this molecule is binding right here on the enzyme. And by binding to that spot, look what happens to the active site. The shape of this area, the active site, is going to change because this molecule binded to it. And it's actually changing in a way, this active site, the shape of it is changing in a way that's going to now make it compatible with binding to the substrate. When that happens, we would call this an activator. So an activator is a regulatory molecule that's binding to this enzyme in a way that's causing it to change shape into its active conformation. So now this enzyme is on and able to bind to substrates and catalyze this certain chemical reaction. And so that's what allosteric activation looks like. The word allosteric, by the way, you guys, allosteric, the prefix allo means different, and then steric means location or site. And so it just means that we have a molecule binding to a different location that's not the active site, and then causing that active site to change shape. And when it's activation, when we're looking at allosteric activation, it's binding to the enzyme and causing the active site to change into its active conformation, its active shape, where it's now able to turn on and start catalyzing chemical reactions. Now the opposite also exists. There's something called allosteric inhibition. And this is the exact opposite. This is when you have a molecule who's binding to the enzyme at a location that's not the active site. And when it binds to that location, it changes the shape of the enzyme's active site, but it's going to change the active site's shape into its inactive conformation, into a shape that's not compatible with binding to the substrate. That's going to prevent it. It's going to change into a shape that's going to prevent it from binding to the substrate. In this case, we would now call this an inhibitor. So this is an allosteric inhibitor that's binding to the enzyme and then causing the active site to change into a shape that is no longer able to bind to the substrate, and now this enzyme's not going to be catalyzing the reaction. This enzyme is going to be effectively turned off because of this inhibitor. Now, when I explain this to students, they always get confused because this sounds exactly like non-competitive inhibition. If you guys remember in topic 2, there's these things called non-competitive inhibitors, and it's basically exactly what I'm describing, where it binds to this spot that's not the active site and then changes the shape of the active site in a way where the substrate can't bind to it. And so for the purpose of this class, think of it as it's the same thing. It doesn't matter. There's no difference between allosteric inhibition and non-competitive inhibition. It's the same idea. And so for the purpose of this class, just think of it as one and the same. Again, that's exactly what's going on. And so this guy is stabilizing the enzyme's inactive shape, whereas an activator is stabilizing the enzyme's active shape, okay? So they're having two different opposite effects here. Now, another thing that students think when I explain this is they always default to think that activators are good. This is good for you. Activators are a good thing, and inhibitors are bad. And inhibitors are a bad thing. They're not good for you. And I want you guys to not think that. Both of these are good. You need both of this happening in your body right now in different cells to be a healthy organism. You need enzymes to be turned on, and you want to stimulate those enzymes to work and work faster and do certain chemical reactions. And then there's situations where you want certain enzymes to not be working and turned on right now, and you want those enzymes to not be performing certain chemical reactions in that moment or in this cell. And so this is all, both of this is super important. Activating enzymes and inhibiting these enzymes are both vital to a healthy organism. You need enzymes to be turned on, doing certain things at a given moment, and other enzymes to be turned off, not doing certain things, and depending on what cells. And that's going to change and fluctuate on a moment-by-moment basis, or depending on which cells it is, or depending on what's going on in your environment. You want enzymes to be turning on or turning off, or working faster or working slower. So think of all of this as good. We need to control the enzymes. Now, another thing I want to talk about in this video is something called feedback inhibition, which is going to come up multiple times throughout this semester. But this is something that takes place when we're looking at metabolic pathways. And so I want to explain to you real quick what a metabolic pathway is, which is also something we're going to be talking about throughout this semester. So this is very often in a very common situation in cells is that there's a certain substance that they have, a certain molecule that they have, and they ultimately want to take that molecule and turn it into this other molecule, right? Like we want to take this thing and we want to turn it into this thing. Now, oftentimes there's not a one and done reaction that can do that, where with one reaction you can take this thing and turn it into this other thing. Many times that's not the case. And so more often what we find is that very commonly, in order to take this molecule that you have in a cell and turn it into this desired new molecule in the cell, there's actually a series of chemical reactions that have to take place in order for that to happen. And it's not just one reaction, but multiple reactions that are going to take place consecutively to take that starting molecule and ultimately turn it into this new molecule that we want. And each of those chemical reactions that it requires is going to be controlled by a different enzyme. And that's what we call a metabolic pathway. So metabolic pathway is when you have a series of chemical reactions taking place to take this starting molecule and turn it into some final product that the cell desires. And so that's a metabolic pathway. So in this picture, you can see that there's this green triangle substance that we're starting with. And in the cell, they want to take this green triangle and turn it into this yellow square because they need the green triangles to turn into yellow squares, and they're doing something important with the yellow squares. But there's not one single reaction that can do that. Instead, you first have to take the green triangle, and there's a reaction that has to turn it into the purple rectangle. And then after that, the purple rectangle, no one really cares about it, but we can take the purple rectangle, and there's another reaction that can turn it into this blue blob. And no one really cares about the blue blob, but now that you have the blue blob, you can do another chemical reaction where you can take the blue blob and turn it into this yellow square, which is the thing that you actually wanted. But that was a series of chemical reactions that needed to take place to ultimately accomplish this goal. And in each of those reactions, there's a different enzyme involved. You had enzyme number 1, and that's different than enzyme number 2, which is different than enzyme number 3, and each of them are controlling each of these consecutive reactions that are taking place. That's exactly what a metabolic pathway is, and that's very, very common inside of cells and lots of chemical reactions. There's a pathway of reactions that are taking place to get one thing to turn into another thing. Now, the reason I bring this up is because there's something called feedback inhibition that takes place oftentimes when we're looking at a metabolic pathway. And feedback inhibition is when the end product of a metabolic pathway, this molecule that you're ending with, is actually able to bind to one of the earlier enzymes used in the pathway. And when it binds to that enzyme, it actually changes the shape of that enzyme in a way where it's no longer able to bind to its substrate. And so that enzyme is now being inhibited, it's being turned off. And now that that enzyme's turned off, this whole metabolic pathway is effectively being shut down because if this enzyme isn't doing its job anymore, well, then the next enzyme can't do his job, and the next enzyme can't do his job, and then there's going to be no more yellow squares being made. And so this thing that I'm describing where this end product in a metabolic pathway is inhibiting one of the earlier enzymes, that's called feedback inhibition. When I describe this to students, they often think, they're confused usually. Students are usually confused by this because they think it's counterproductive. Like why, like your initial thought might be, why would the thing you're making want to turn off one of the enzymes that was used to make it? Like this doesn't seem productive at all. Like you're making this thing just to turn off the thing that was allowing that thing to be made. And really though, this is actually really smart for cells to do this. And the reason why is because this feedback inhibition would only be happening, or would be happening a lot, if there's a lot of this end product being made and it's starting to accumulate in the cell and no one's really using it. So imagine a situation where, yeah, we want to take the green triangles and we want to turn them into yellow squares because the cell needs to use the yellow squares to do something important that it wants to do with the yellow squares. Well, then this metabolic pathway can be used to do that. But let's say that now there's a situation where you're taking all these green triangles and you're turning them into these yellow squares, but no one seems to be using the yellow squares. So these yellow squares are just starting to accumulate in the cell and no one's using them. And now you're getting to a point where you're kind of wasting energy and you're wasting resources doing something that apparently no one cares that you're doing. So at this point, you're taking all these green triangles, which maybe you could be doing something else with those green triangles, and you're making these yellow squares, which apparently you have a lot of now, and no one seems to be needing them. And so what's going to happen now is once you have a lot of these yellow squares floating around, by coincidence, because they're just floating around, all this is floating around in a cell, some of them, if there's a lot of them, some of them are going to start binding to this earlier enzyme and turn off this enzyme, which is going to shut down this pathway to stop more yellow squares from being made. which is actually a perfect solution to this problem now. We had too many yellow squares, and now this yellow square is going to start binding to and inhibiting this enzyme, which is going to prevent or lower or decrease the amount of yellow squares being made, which is actually very efficient for the cell. It's going to prevent the cell from making too much of something when it doesn't need it. Now that's temporary. At some point, the yellow square will fall off. If you guys remember these inhibitors, they don't last forever. They bind, they inhibit, and then sooner or later they fall off. And maybe minutes later or hours later, there's not a lot of yellow squares. And so the yellow squares are starting to dwindle down, and now the cell needs more yellow squares. Well, sooner or later, like this pathway will turn back on when there's not a lot of yellow squares. If there's not a lot of yellow squares, they're not going to be probably binding. It's going to be very unlikely that they're binding to this enzyme. And so when there's not a lot of them, this pathway is probably turned on. And when there is a lot of them, this pathway is going to be turned off, which is going to help cells control and regulate how much this, when this metabolic pathway is being used, and make sure that it's not being used when you're starting to become wasteful. So I hope that kind of makes sense. We'll see lots of examples, or a few examples of that in the future in this class. Here's another example of that real quick, though. I won't spend a lot of time on this, but this is just showing you a real example where there's a metabolic pathway that takes this, this molecule called threonine, and there's a series of 5 reactions that turn it into this end product called isoleucine. Each of those reactions involves a different enzyme, enzyme number 1, enzyme number 2, 3, 4, 5, to ultimately get this guy to turn into this guy. And when there's too much isoleucine being made, too much of this molecule being made, you can see that it actually can start binding to this first enzyme and start inhibiting this enzyme so that it's no longer able to do this first reaction, which is going to shut down the rest of these reactions, which is perfect. That's exactly what we want. When there's too much isoleucine, and when there's so much of it that it's starting to bind to all of these enzyme number 1s and turning them off, well then yeah, we probably should start slowing down and turning off this metabolic pathway because we have too much isoleucine. And when there's not a lot of isoleucine, if the cell is using a lot of isoleucine and it's using it quickly, well then this pathway will probably stay on because we're going to make isoleucine and it's going to be used up and it's not going to accumulate in the cell, and it's not going to get to a point where it's going to start inhibiting this earlier enzyme. So anyway, that's what's happening here. And then the very last thing in topic 3 here is the fact that in cells, these enzymes are localized to specific areas inside of a cell. And so inside of a cell, cells are very complex. We haven't talked about the structure of a cell yet. We will in Unit 2, but hopefully you kind of remember cells from your freshman bio class. Cells oftentimes have these different compartments and areas inside the cell where different things are happening. And these structures have different functions inside of the cell. We call these organelles, and we'll talk about all this later. But basically what I want you guys to know right now is that cells compartmentalize where certain reactions are taking place, meaning that different reactions take place in different areas of the cell. And so depending on the type of reaction you have, that type of reaction is taking place over here in the cell, and this other type of reaction is taking place over here in the cell. So it's all being compartmentalized. Like this stuff happens over here, this stuff happens over here. And this is good because it increases the efficiency of the cell by preventing chemical reactions from chaotically happening everywhere, or from competing with one another and preventing each other from working. So it helps to organize all of this and make sure cells are operating efficiently. And the way cells are able to accomplish this is by making enzymes. So the enzymes are the ones controlling the reactions. And basically inside of a cell, what you're going to see is that certain enzymes are going to be found in certain areas of the cell, and other enzymes are going to be found in other areas of the cell. And because there's different enzymes in different parts of the cell, those different parts of the cell are now going to have different reactions taking place, because it depends on what enzymes you have. That's going to affect what reactions are taking place. And so in cells, enzymes are often incorporated into specific membranes. So a lot of times you'll see these enzymes embedded in different membranes found inside the cell, or sometimes those enzymes will be floating around surrounded by these different membranes that we find inside these cells. And so by having these different compartments in a cell, compartmentalizing different enzymes and making different enzymes be found in different spots in the cell, that's going to allow a cell to have certain chemical reactions happening in certain areas and other chemical reactions happening in other areas. So an easy example of this is ATP synthase. I talked about this enzyme briefly in topic 1. This is an enzyme that makes ATP. Its job is to synthesize ATP. And this enzyme is basically found in the mitochondria of the cell, in the inner membrane of the mitochondria. For now, just know it's inside the mitochondria. This enzyme is found embedded inside the mitochondria. And that's basically the only spot that we find this enzyme inside the cell. It's inside this special organelle, this special structure called the mitochondria. And so that's where ATP is going to be made. We're going to have a bunch of ATP being made in this area of the cell, the mitochondria, because that's where the enzyme is that makes ATP. So ATP synthase is the enzyme that makes ATP, and we find this enzyme in this specific structure inside the cell. So only in that area will that reaction be taking place. And that reaction is not going to be taking place in other parts of the cell. And so just conceptually know that inside of a cell, there's different reactions taking place in different areas because different enzymes are localized to different areas of the cell. But that's it for topic 3 and for unit 1. We made it to the very end. Congratulations, you guys. I'll see you in class. Thank you.